Hello dear all beautiful students, welcome in for biology class. Once again the weekend, last class of this weekend. We are connecting here and welcome to this class. And biology first paper, in very last class we thought about the plasma membrane, about their function and some model. Today we will go through a very important topics like fluid mosaic model, okay, on the basis of the cell membrane or plasma membrane. That is very important, okay, for CQ types of question. Fluid mosaic model, it is on the basis of the cell membrane. What is the concept from come here? Cell membrane or plasma membrane. On the basis of that concept, the fluid mosaic model is very established and very important. In 1972, Singer and Nicholson, okay, S.J. Singer and G.L. Nicholson. S.J. Singer and G.L. Nicholson is the founder of this model. In 1972, they gave the acceptable fluid mosaic model that is very important for you in any types of term exam or public exam also. Very important model. We will try to understand this one with drawing the figure very first time. Okay, you can draw with me. I will try to draw like this way. I just draw shortly. You can draw by following me here. I can draw in this way and just facing off like that figure. This way we draw the figure very first time, and as like as the previous time we can ensuring this one. You can draw the figure very first time, and firstly follow me so that it will be very easily for drawing for you, and better of drawing is better of good mark carrying out chance, okay. So important, this one is very important for drawing the figure, and facing will be there. In very last class I think you got some of the important points about the membrane concept. Today we are going through understanding about, you draw with the pencil, please don't use the pen, draw with the pencil. That will be the better experience of getting this one. Yes, and finally I can break out from this part. Where is the duster? Yes, this one I will just erase two of this part and draw a figure like that here, so that I tell you to draw it with the pencil. I just draw this one in this way, and having the part like that. Please follow me, my dear students, follow me. This one, and I can draw this one also in this way. This way we can try to draw the figure, okay. I will mention the indication words. You can follow me first for drawing the figure. This type of a structure we can see, and also having, I can take out some one of this one from there for drawing the structure like that, and with having the features of this one. Try to draw and follow me, my dear students. I hope so you are getting well and fine, and you are connecting here by drawing the figure, because in biology drawing figure is mandatory, because a figure carry out the maximum number of marks if your figure is not getting well, and if you are not getting used to about drawing figure, that times you cannot carry out the optimum marks or good figure in the exam. Okay, so it is very important to you. And I can another one I can draw here. In this way, I will make sure and marking the part also. Please at first draw this one, and then we can you can spread the drawing figure in long page or bold figure, but since my board has the consisting and congesting parts, so I draw it in short way. Okay, the lengthen of this size. In this way we can draw the figure. Very important of drawing figure. So this is the inner side or inner surface of the cell. Inner surface of cell, and this one is the outer surface. Outer surface of cell. Outer surface of cell, you can write this one, and these are the cell membrane. Actually what we deserve for cell membrane. Yes, and this is the integral protein. This one is the integral protein, and this one is the peripheral protein. Yes, and sometimes in between them another thing you can draw, like that type of a structure. Okay, you can draw that type of a structure in between them. These are the cholesterol. Yes, and these are the lipid bind protein is also there. These are known as the lipid bind protein. This one also lipid bind protein. Yes, then this is the glycoprotein. This one glycoprotein. This one is the glycolipid. And this one are known as the phospholipid bilayer. Okay, this one is the phospholipid bilayer that we learned in yesterday about drawing this one, and hope so you understood about marking that parts. And these are the cytoplasm we can write, and figure fluid mosaic model. Very important question for CQ types of question asking here, and I hope so you will getting fine for drawing this figure. Okay, I have seen that many of you are joining there. I can see there. Afra Ahmed, Minhaz Uddin Ahmed, Naurin Siddiq, Charles Shen, Labiba Alom, Rahul, Ahanaf Tajwar, Abrar Chaudhuri, Amina Ifrat, Raisa, Aditi Mahajan, Muhammad Ashraf, Jarib Tajwar, Samia Afroz, Soha. So all of you are welcome and very good morning to you all, and I hope so these topics will be very helpful for you for understanding this one, because drawing figure is very important for particular question in case of the biological science. And you know that biological science is nothing but very important for your future and brightened future. Like that, if you want to get wish to be a doctor, if you want to get admitted in well renowned university, so any case the biological subject is very important. And not only for this one, any job recruitment, there are written part is also very important. Any case, suppose now you will think your future, okay? So drawing figure is very important, and we have drawn the figure, and this is given by 1972 S.J. Singer and G.L. Nicholson. And I draw the figure in a space, so I get some space to write something about this one, okay? So this model actually double layered, okay, double layered. According to this concept, the cell membrane constituents double layered membrane, we can write very first time double layered membrane, and each layer consists of phospholipid. I tell you, cell membrane, this is the part of the cell membrane, and this is the outer surface of the cell, and this is the inner surface. Since the cell membrane is there, outer surface and inner surface, we can see. So this layer is actually phospholipid bilayer. That means each layer, this one is the one layer, this one is the second layer, okay? First layer and second layer consist of phospholipid. Very important points of them. Yes, and here hydrocarbon tail placed face to face. I will make you understand very easily, please connect here. And hydrophilic head placed in opposite side. So what I have written here, my dear students, this is the double layered membrane. Each layer consists of phospholipids, so that phospholipid bilayer, this is named as. And there I told you yesterday that is hydrophilic and hydrophobic. And hydrocarbon enriched, these are also hydrocarbon enriched tail, or hydrophobic, you can say like that. They can face to face, this layer face to face they are placed in, but the head, hydrophilic head, are placed in opposite side. So this is the first concept of fluid mosaic model, we learn it here. Other basic constituent according to this model, the structural constituent we will try to understand now. I hope so you understood this one. If any problem you face, please knock me there in the comment box or anywhere. If you getting any asking question in your mind, then I will try to make you understand, okay? So this one is actually a very important one, and then now I will write the second step about that. So I think you are collecting this one perfectly. This is very important amongst all the topics. Most important topics we learn here now. So this one is very important. The structural constituent of cell membrane, according to this model, structural constituent of cell membrane or plasma membrane, you can write. Number 1, phospholipid bilayer. What we have written, phospholipid bilayer. phospholipid bilayer. Okay, so bilayer means it is double layered. Phospholipid bilayer is the first one, double layered. Okay, and then we can see each phospholipid that means the phospholipid bilayer, each phospholipid equal to 1 molecules glycerol, 1 molecule glycerol plus 2 nonpolar fatty acid tail plus 1 polar phosphate head. I write somehow small with complete of writing in there, so that okay. So what I have written there, my dear students, look at here. This is very important things I have written. I hope so you are getting well also. Look at here. Later on I have seen many of you also joining, Ayan Sami, Milon Fanny. Okay, well and good. I want to say what the important things. Phospholipid, first one, structural constituent of cell membrane. This is the cell membrane. First one, phospholipid bilayer. Seen, this is the one layer and this one is the second layer, and each phospholipid what contain 1 molecule glycerol. One very important, I write in very short way. Each phospholipid layer consists of 1 molecule glycerol, 2 non-polar fatty acid tail. You can see this one is the 1 and this one is the 2. That means 2 non-polar fatty acid tail plus 1 polar head. Okay, so each phospholipid bilayer, we have seen this is the phospholipid bilayer, means 2 layer. Each phospholipid contain 2 non-polar fatty acid tail and 1 polar fatty acid, phosphate head, and in between them there is nothing. We have seen in between them the glycerol is 1 molecule glycerol are placed in them. So I think you understood this one, that phospholipid bilayer, if it is going on there, so it is double layered, and this phospholipid is phospholipid non-polar fatty acid tail and polar fatty acid, phosphate head, and in between them there is glycerol, 1 molecule glycerol are placed on. So phospholipid bilayer, first part and first one, I think you understand this one. Second constituent. Now we'll go to the second constituent. Okay, let's go. Membrane protein, number 2. Membrane protein. Okay, membrane protein, there are 3 kinds of protein. Kinds of protein are found here. Membrane protein, 3 kinds of protein are found. First one, integral protein, and this is placed actually, where it is placed, in both surface of membrane. It can be found. Okay, integral protein, look at it. This is the integral protein. This is the lower surface, this is the upper surface, this is the inner surface, this is the outer surface, and both surface they are placed in. Both surface of membrane, integral protein are found. Peripheral protein, and this peripheral protein, the lower surface of the membrane, lower surface or inner surface, you can write lower surface of membrane. Placed in, you can write the placed in lower surface. Look at here, peripheral protein, though the integral protein in the outer surface and inner surface, both surface they are placed in, but the peripheral protein just lower surface or inner surface of the cell membrane are placed in. And number third, lipid bind protein. Lipid bind protein. This lipid bind protein placed in lipid core. Lipid core. Lipid core. Okay, look at here what I have written there. Very importantly, these are the very important but easiest way I make you understand, so that it will be best for learning of you. What actually mentioning here, lipid bind protein. Lipid bind protein, sometimes I move backward the mobile phone that it look like blurry or clear vision. Sometimes the board is looking blurry on that case. So lipid bind protein placed, look at here, lipid bind protein placed on the lipid core. Where the phospholipid are there, the lipid bind protein can placed on the specific core. Okay, and so they are also membrane protein. Lipid bind protein, since the membrane layer they are placed in, so they are counted as the lipid bind protein, and lipid core they are placed in, so they are membrane protein. They are also known as the membrane protein. Lipid bind protein, we can say the lipid bind protein can also termed as the membrane protein. Okay, I hope so you understood this one. And number third, glycocalyx. Number third is the glycocalyx. 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 Yes, it is very important that it is a sugar layer. Sugar layer. Glycocalyx. I don't give you the definition because the glycocalyx is very important for asking in the exam. Yes. Glycolipid and glycoprotein. Protein plus glycolipid. Glycoprotein and glycolipid to bind together is called glycocalyx. Okay, so glycoprotein how can form there? Look at here, I have written there the glycoprotein. Look at here, this one, glycoprotein, and lipid is also there, glycolipid. Look at here, this part is termed as the glycolipid and this one is termed as the glycoprotein. How the glycoprotein form? Glycoprotein can form with one molecule carbohydrate with the protein, lipid bind protein. Carbohydrate plus lipid bind protein complex together to form the glycoprotein, and this glycoprotein when bind with the glycolipid, both types, glycoprotein plus glycolipid together, we can say the glycocalyx. It is asking for 2 marks question or 1 marks question, what do you understand by glycocalyx? So on that time you will write the combination of glycoprotein and glycolipid is called glycocalyx, and it is a sugar layer, and we can say here the lipid binded protein when connecting with carbohydrate chain, then glycoprotein form. This glycoprotein and glycolipid to form glycocalyx, very important thing also. Okay, then last part is the cholesterol. So this one you can see there, this one is the, we have seen in between them, this one we see in between that one. It is cholesterol is actually lipid enrich or fat enrich, you can write enrich material. Lipid enrich material. We can say they can, it is placed in between in between phospholipid bilayer or phospholipid, you can write phospholipid. Okay, yes, and it can be changed, due to cell surface permeability. I write very short way about the fluid mosaic model, and this model is very important one. So last point was cholesterol. You have seen the phospholipid bilayer. In between them, sometimes this type of structure we have seen some lipid enriched materials or matters, and these are known as the cholesterol. And these cholesterol, when we found in between the phospholipid bilayer, what is the variation in them? They are placed in a number of the cholesterol on the basis of the dependency due to the cell surface permeability. If the cell surface permeability is more or less, so the position and place and number of the cholesterol will be also vary from the position, place and numbers also the cholesterol. So this is all about the fluid mosaic model of cell membrane. That is very important question for CQ. You can search in any previous year question or any types wanted type of question is fluid mosaic model. I draw the figure very first time and try to make you understand what is going on there, the outer surface, inner surface, phospholipid bilayer, in between what types of protein and where it is placed in and what types of constituents are in there. We understood this one. I hope so this learning will carry out a good understanding for your next preparation. That's all about today, my dear students. Stay safe, sound, stay connected with the study. Allah Hafiz.